Esteemed PhD candidate, the College of Deans of this university, represented by us, has read your PhD thesis and the propositions that come with it. And we are ready to hear you defend them. Please be seated. Stay on your feet.
I'm going to make sure of that.
Please. Does anyone in the audience wish to exchange ideas with the candidate? I see that this is not the case, and I therefore call upon Professor Faber from the University Medical Center here in Groningen, the Department of Gastrointestinal and Liver Diseases, for the first opposition. Professor Faber.
Thank you, Mr. Rector. Geachte mevrouw de promovendus, dear candidate, I think I will continue in English. First of all, congratulations with the completion of a very nice thesis, I must say, very impressive, a very thick book. And as you know, that my research is also focused on liver and intestinal fibrosis. I must say I read it with much more, or even let's say more than normal interest, especially because there is a lot of hot new data in there which is not published. So it's always good to have the feeling that you are the first one to read this. So that was really nice. And of course, your work is let's say, continuing on a long tradition of working with tissue, precision cut tissue slices. And I also have, let's say, read, of course, many theses of the ones that preceded you, and also their propositions. And there is, I think, always one specific proposition that comes back always. So, and that's number one in your case. And I would like one of the Paranymphen to stand up and read this first proposition out loud to the public.
Precision cut tissue slices contribute to the understanding of how certain laboratory animals fit into drug development.
Thank you very much. So I think there is not much to argue about this, and I think that propositions are there to have a little argument, to tickle the brain a little bit. So I want to come up with an alternative proposition for you, but I want to introduce it also, that you know what the background of my alternative proposition is. So I want to go to chapter 2, where you did this RNA sequence analysis. I think this is really a very crucial and important next step for, let's say, the tissue slice research, to determine how they actually behave in culture. Well, there you take, let's say, kidney, liver, jejunal, colon, ileal slices from mouse and from human, from before and after incubation. So it's a really big matrix of different conditions, and you compare them. And then what surprises me in the end is basically shown in figure 2, panel E on page 33. Is that if you look at the effect of culturing, it follows the species. It does not follow the organ. So that's remarkable. So yes, human kidney slices behave similarly as human liver slices. So here comes my alternative proposition. My alternative proposition is human kidney slices, yes, are a better ex vivo model for human liver than mouse liver. I would like to hear a direct yes or no and an argumentation.
Okay, highly learned opponent. First of all, I would like to thank you for coming to my defense and for your nice words about the thesis. And indeed, if we're looking at figure 2E on page 33, we do see that the response of slices during culture...
Whether it's yes or no?
No.
So then we know what the state is, yes.
If we look at our results, we know that slices during culture and after the preparation have a non-specific response of inflammation and tissue remodeling. this non-specific response is characteristic for each species. So that's why I consider that the slices are, like human liver slices are similar to kidney slices regarding the non-specific response. However, if we're thinking about the cellular composition and the tissue type, I do not believe that liver slices, human kidney slices can be more representative for human kidney slices than the mouse ones.
Okay, so you consider whatever you see now as a non-specific response?
Non-specific, but like it's a non-specific response, inflammatory and tissue remodeling response, but each organ showed different patterns.
But how would you then filter out the specific response? Because I do not think that I read anything about the specific response in your results.
Actually, if we are going to page 44, on figure 8, we did a 4-set Venn diagram that is illustrating actually all the changes in both species and in different organs. So for example, I am more interested in liver. So here we can see all the differences between each organ, and we can see the differences between human and liver. And if we want like a more detailed analysis of these figures, we can go to supplementary figure.
Yeah, I think that the figures will become too small actually.
Actually, I don't know. Yes, it's a file and it was too big to put it in. But for example, we want to see which pathways are changed only for human and not for mouse. And then we check, for example, group D and C, because these are going to be pathways altered in human and not in mouse. And we will consider, okay, we cannot study these pathways in mouse because we will not have a similar response, so then it would be just...
Yeah, so but let's say even the specific response, I think, are mainly buried within, let's say, an overall response of inflammation and probably wound healing, fibrosis, right? There are only very details if you look at, for instance, liver and bile acid metabolism or something like that, which is a specific function, and you could look for cholestatic liver diseases. I think that your promoter already did this in the past. So how is that actually? I think that there is very little information on that, right?
Yes. But I don't know if I see your point.
Well, the point is that, let's say that you basically now looking at a very general effect of the culturing, and I think that the non-specific effect, as you call it, is really overarching many of the inflammatory pathways, that it is very difficult to extract the specific pathways that you may be interested to study.
Okay, now I understand. So indeed, the inflammatory processes are there. So if we are interested in a pathway that, for example, is downregulated by inflammation, we could actually culture the slices with some anti-inflammatory compounds to stop or reduce the inflammation in order to look better at the pathways that can be directly influenced by inflammation. So we can adapt the culture conditions to have a better view of the pathways that we're interested in. So that's why here we have all the pathways, but they are mostly in the supplementary files. So if a different investigator has a different idea on a certain pathway, it has to go to the supplementary files and can see what's happening to these pathways in the regular conditions, and could try to see like if anti-inflammatory compounds cultured with the slices can help that pathway, for example, to not be inhibited, and so we can play around.
I understand. I already got a signal from the director, so I'm very sad to say that we have to stop this discussion. Fortunately, I have many more difficult questions present for the next opposition. So I'm happy to return the word to the director, and I look forward to the next opposition.
Thank you, Professor Faber. It's then my pleasure to invite for the next opposition Professor Jimenez, who is coming to us from the University of Barcelona. Thank you for traveling all the way to Groningen, Professor Jimenez, and the floor is yours.
Thanks. Thank you very much. Thank you very much. First of all, I would like to say that I'm very pleased to be here, and also to thank to the candidate the excellent work that she has performed. In particular, because we, in my lab, we are very interested in this subject, and we have been looking for this kind of solution for a long time. In particular, because we want to solve the problem in the transition between the animal models and the humans. And I think that your work is pioneering this kind of problems. Then I just want to start with a question that is rather than to be focused on the thesis, is much more, from my point of view, of conceptual. So you and maybe we need to convince to the other scientists that this is a real model of fibrosis, and that you can translate the results that you found in the slices to the humans and the results that you obtain. However, the circumstances, in spite that you find very nice results, are quite different from my point of view. And then this is an objection that probably will emerge in the next year, particularly to you. That is, the liver disease that ends in fibrosis, in a fibrosis activation process, it's in general, in most cases, a process that takes many years. It's even in some cases 30 years, 40 years. And here you develop a model that an advantage is that it's very quick. But at the same time, one's asked to himself whether or not this is the same circumstances that the conclusions that we obtain can be extrapolated to humans. So I would like that you discuss all these considerations, because I am sure that you spend some nights thinking about this problem.
Highly learned opponent. First, I would like to thank you for being part of my reading committee and for coming here to be the opposition. And now I will address your question. So indeed, when we think about fibrosis, it's not a process that can happen in few days, in few months. It actually takes decades. So then the question is, can we convince scientists to use a model that takes 2 days that this is relevant? But that's why we're using not only healthy tissue, but also diseased tissue. And we consider we have 2 types of model. So for example, when we have healthy tissue and we slice them and culture them, they are developing fibrosis, but we consider that the early stage of fibrosis. So it's an early model. But it's very important that we use also diseased tissue that comes from patients who have cirrhosis, and then the extracellular matrix is present, because this extracellular matrix is not just inert, so it also produces other mediators that are influencing fibrosis. So we do consider this a great model for the late stage of fibrosis. So if we want to test a drug, we will always try to test it in the early stage, that comes from healthy tissue, and in the late stage, that comes from diseased tissue. And based on that, we can see exactly like if drugs work better in the early stages or in the late stages. However, we do have to consider that this is a model, so of course there will be differences to the in vivo situation, but we do consider this being a great first step in verifying the efficacy of a drug. So we are not going to replace all the models with our model, but we think that this can contribute to drug scanning and accelerate the drug discovery process. So I hope this answers your question.
Yeah, thank you. There is have more time, so.
Yes, certainly.
Thank you very much.
About 4 minutes.
Yeah, a second question that is much more methodological. I'm a little bit worried because your incubation is made with 80% oxygen. And then I'm not pretty sure of 2 things. Well, the first question is, or the question, why you use so high levels of oxygen? Because from my point of view, this could induce some kind of oxidative stress that would be away from the general condition of the experiment. So could you justify this concentration of oxygen in your cultures?
Yes. So for example, each organ is different, but for liver, if it's incubated similar to cell culture at 20% oxygen, this will result in the necrosis of the tissue. So we know that liver doesn't, if we want to culture liver slices, we do need 80% oxygen. I do understand your concern regarding oxidative stress. We did measure in the past if there is high oxidative stress in the slices, and there is a bit, but just a bit, so not sufficient to justify not using 80% oxygen. We also tried to measure glutathione and also the ratio between reduced and oxidized glutathione, but there were no changes. But this depends on the organ. So for example, if we're looking at lung, an organ that is more exposed, that in our body is exposed to the atmospheric oxygen, indeed, if we culture a lung at 80% oxygen, there is high oxidative stress, and there is more apoptosis induced than when the lung is cultured at 20% oxygen. Therefore, it's really dependent on the organ, the type of condition, the culture condition.
Okay, thank you very much. It's very clear the answer. From my point of view, is enough. Thank you.
Thank you very much, Professor Jimenez. It's then my pleasure to invite for the next proposition Professor Bank from the University Medical Center, where he works in the section Medical Biology. Professor Bank.
Okay, thank you, Madam Director. Esteemed candidates, first of all, I want to congratulate you for this very nice thesis, and also I want to congratulate your supervisor, co-supervisor. I also like very much your cover. There is a hockey stick over there, and we all know that fibrosis is a very devastating disease, and there is no real effective treatment against it. So if you have an effective drug, I think, let's say, the business model will increase like a hockey stick. So therefore, I like it very much. I have a question with respect to chapter 6. There you have investigated a particular gene, GPNMB. MB, perhaps medical biology, I don't know. It is highly increased in diseased liver, about 1,000-fold or something like that. Is this really an interesting marker, yes or no?
Highly learned opponent. I would also like to thank you for being part of my reading committee and for being here today. I do think it is an interesting molecule to investigate, also because its role is not elucidated and is present not only in fibrosis but in different pathologies. But I think more research should be done to investigate its exact role, because in certain studies it can be, for example, it's very expressed in melanoma cells and can have a good effect or a bad effect on cancer cells. But for liver, it's still not clear, because it can also act like an anti-inflammatory compound. It's reducing the expression, anti-inflammatory protein. It's reducing the expression of IL-6. So I think to a certain level it can be beneficial for the liver, but we don't know if, since the diseases will advance, if it can be detrimental to the liver.
Okay, so you're talking about a little bit about the function of the gene itself, indeed. But if you have an antifibrotic drug like elafibranor, for example, you showed in your thesis that if you use that, there is no change at all in this biomarker. It remains the same, it remains stable. Is it still an interesting marker then?
Yes. So we did look at elafibranor, but we also checked the effect of elafibranor on collagen and on other fibrotic markers, like the chaperone HSP47 and fibronectin. And elafibranor did not have any antifibrotic effect, so it did not reduce these markers. So what we see in slices that 48 hours culturing with elafibranor does not affect fibrosis, and it also didn't affect GPNMB. So I do believe, we also try to assess the correlation between collagen and GPNMB, and besides our culturing, it was moderately or strongly correlated. So I was expecting to not see a change in GPNMB expression by elafibranor, because we also didn't see any effect in the fibrotic models.
Okay, I'm looking forward to the functional, let's say, properties of this gene indeed. Then you also mentioned a little bit in a general discussion about markers of collagen synthesis and collagen degradation. Collagen deposition is the hallmark of fibrosis. What is actually the best marker, the synthesis marker or the degradation marker, in your opinion?
Well, I would say if the patient is not on a treatment, then I would say it's important to evaluate synthesis markers to see if the disease is progressing. But if the patient is following a treatment, then I would say we should check the degradation markers to see if indeed the treatment is leading to the degradation of the extracellular matrix.
So, okay, I agree with you. Let's say the synthesis marker is the most promising one indeed, because I mean, there's hardly any degradation of collagen going on. It is irreversible, so I don't expect much of a degradation marker. Let's take a look at chapter 3 on page 80. There you show a table on page 80, and you showed actually several pathways. And in the kidney and in the ileum, the first pathway that you mentioned is the osteoarthritic pathway. And that I don't understand. In the kidney and the ileum, I mean, cartilage, osteoarthritis, it is a very different tissue with different extracellular components. So, what is actually up or down regulating in this so-called osteoarthritic pathway?
So, the osteoarthritis pathway is a pathway that is composed of genes that are involved in tissue remodeling, in inflammation, in matrix degradation. So it's a very general pathway, but of course we don't have this type of cells present in the kidney or in the ileum. But what we see during culture is a lot of inflammation, a lot of tissue remodeling. So because they are sharing these genes, the pathway appears as activated. And we believe that the cells involved in this case are the fibroblasts and the immune cells that are secreting this inflammatory, and for example we have MMPs and all types of collagens that are involved in those osteoarthritis pathways, and these are also the genes that we observe that they are changed during culture. So because we have this ongoing inflammation and a fibrogenic response, this is common with the osteoarthritis pathway, so that's why the pathway is appearing as being activated.
Okay, so you are apparently talking about inflammation pathways in osteoarthritis and not, let's say, with the collagen, because the collagen in cartilage is quite different compared, let's say, to the ileum and the kidney.
Yes.
Do we still have time for another question?
If it's a short one.
It's a short one. Okay. Let's take a look to page 85, the table over there. There we see, let's say, difference between the disease and the healthy tissue slices. And the A means it is the predicted pathway activation, and the I means there is inhibition. Now, first of all, I don't see an I, only an A. But you see, let's say, the colors and the A, there is a big discrepancy between what is predicted and what is actually seen. So, are all the predictions that we have made wrong in the past?
No. So first, I would like to explain how we thought this pathway, this table. We wanted to show our pathways of interest, and the engineering pathway analysis is offering us two different statistical analyses. The first one is the p-value, which is showing if, in general, the number of changes observed in the pathway are statistically significant, while the activation and inhibitions are based on the upstream regulators that are dictating if the pathway will be activated or inhibited. So we are looking at two different statistical methods that are showing different things. So yes, we can see if a lot of genes in the pathway are changed, or we can see if the upstream regulators are dictating if this pathway should be activated.
Okay, so I conclude actually that my interpretation of this table was a little bit wrong. I give the word back to the rector.
Thank you very much, Professor Bank. And it's then my pleasure to call upon Professor Groothuis from the Department of Pharmacokinetics, Toxicology and Targeting of this university for the next exposition. Professor Groothuis.
Thank you, Mr. Rector. Dear candidate, I'm very happy to be here part of your examination committee, and I would like to congratulate you for this really, really good thesis. I read it with a lot of interest, you know why, I suppose, but I want to include also your supervisor, your co-supervisor in this, but also your partner, because you did a lot of work together, so we will talk about that later. But I know how much work this is. It sounds very simple to say, okay, liver, intestine, and kidney from human and from mouse, and it's in one chapter or 2 chapters, but it is an immense book. So congratulations. And I a little bit expected this of you, because I was your supervisor during your master, and I was your mentor, and already that time you behaved like a PhD student, and people often confused you with your supervisor, PhD student, as you being the supervisor. So I think my expectations came out. Okay, I have a few questions, and I will start with a more general question. What I do see in your data, in what happens during incubation, and that there is no information whatsoever about the genes involved in transport and metabolism. And I know that in the medium that you used, there is a strong downregulation. At least that is what everybody else has found. So I would like to ask you in the first place, did you see that in your data? Or did you not see that in your data?
Highly learned opponent, thank you very much for your kind words. So we did see in our data a lot of downregulation for transport and metabolism, as you mentioned. We also compared the data with previous data published on the subject, and we noticed that in the first 24 hours there were not reported so many changes in transport and metabolism, but we do see after 48 hours a lot of changes. And yeah, like transporters are one of the top genes that are being downregulated. But I don't know if I got correct your question about inflammation regarding to this.
No, I didn't say anything about inflammation yet. There's lots of data to show that if you use these slices for drug metabolism studies, that after 24 hours it decreases very rapidly, which is a problem in studies on that topic. And I think it is important because you want to use this model to study the effect of drugs. And drugs in the body are generally rapidly metabolized, or they come into cells by transporters or exported by transporters. I mean, you know all this. So is your model then a correct model to study the effect of antibiotic drugs?
Well, I would believe that it is a good model to study antifibrotic drugs. Also, we are using higher doses, a bit higher doses, because we know that in time, with the decrease of transport in the cells, the cells will be less exposed to the drug. We also have a limited incubation time, so we do use only 48 hours. But we noticed, for example, if we look at chapter 4 and chapter 5, we did see the effect of antifibrotic compounds. And if we compare these antifibrotic compounds with results seen in animals, in vivo, or in cell culture, or even in patients, we do see similar effects. So I do believe that even with the downsides, it's a good model.
Okay. And why did you choose for this incubation medium? Because there is an incubation medium which retains these functions more to the original level. There's a medium which shows only 700 genes changes after 5 days, whereas in your case there are 4,000 genes changed after 2 days, which is really huge. It's a quarter of all the genes, I think, that is changing. It's not only inflammation and fibrosis, but there's probably a lot more going on.
So indeed, there are different options for the medium. We did choose this one because we are trying to use slices as a model of fibrosis and inflammation, and with this type of medium we do see these changes. So if we have less inflammation, less changes, we assume that there might be a change also in the fibrotic response. There is also a problem that the medium that you mentioned, I think you're referring to the Celartis medium. It's very expensive. So there are also practical reasons for choosing this medium, and also we don't know the exact composition of that medium, so it would be very hard for us to reproduce it. But the results that we have until now with our medium are satisfactory, and we can see the effect of the drugs. So we do consider it good. Also, my colleague Makarnev did a study on different types of medium and on viability, and indeed the Williams Medium Mini showed good results. So we think it's a good medium for culture.
Okay.
A very short question and a short answer, please.
It's maybe not a short, the question can be short, but the answer is difficult, but just give it a chance. For the future, what we would like to have is a body on a chip, if we call that. Several organs on one chip and study a more integrated system. Do you think it's possible with slices? Because what we see is that for different slices, you use different media, for instance, different antibiotics and so on. Would it ever be possible? What do you think?
Yes, I think would be possible. Maybe, well, we have to look in all the possible antibiotics that are not affecting the drugs. Maybe using working sterile instead of semi-sterile, so then we have less contamination problems. Although the intestine, I think it's quite hard to clean and make it sterile, but I think it is possible.
Okay, well, let's hope for the future. Thank you very much for your answers.
Thank you very much, Professor Groothuis. I then call upon Professor Van Goor from the Department of Pathology at the University Medical Center here in Groningen. Professor Van Goor.
Thank you, Mr. Rector. Dear candidate, congratulations with your thesis. It's really beautiful, and like the previous speaker, I agree. I see that it is an extreme amount of work. You worked very hard, and I also want to congratulate your mentors. You actually say in one of your propositions that hard work is necessary, but it's not enough. Immediately brought me back to a conversation I had with my friend from high school, my only friend from high school. He became a multimillionaire. I was of course complaining that we had to work so hard all the time. And then he said the remarkable words, Hadi, working hard will never make you a millionaire. I was thinking about that. So what else do you need? And you can only pick one. Luck, intelligence, social qualities, or choosing the right mentors.
I learned upon it. Well, thank you for your questions. Well, if I would be politically correct, I would say choosing the right mentors, but I do believe it's luck.
I knew you were trying to escape. You have a great model, and the model brings you close to humans, and it avoids the use of laboratory animals, which I very much like. The only problem I have with the slices is that I would like to culture them for a longer time, and you also want that, a week or 2 weeks, because then you can see changes in the sections you make afterwards. If you would have time to work on that, I mean, a lot of people work on that, and you're always thinking about it. What are your thoughts? Where should we go from here to increase the culture time to 1 or 2 weeks in order to see better changes in tissue sections after the culture time?
Well, I would consider that we do have to work on the inflammation and reduce it, because all these inflammatory markers can activate apoptosis, and all the cells that are dying are transmitting further messages to the other cells. So I would say we do have to act upon the cells that are appearing activated, especially the ones described in chapter 2 and 3. But then we have to consider that maybe our fibrosis model can be affected because we have less inflammation. We might have less smaller fibrogenic response. So to culture it and maybe like slow down the degradation processes, I would consider we have to act mainly upon inflammation.
Have you tried it to reduce inflammation in your slices?
We did actually, not me personally, but we did have colleagues who worked on the reduction of IL-1, and if I remember correctly, I think it had good results.
That brings me to the next subject. You were already talking about oxidative stress. Reactive oxygen species and sulfur species can also damage your organs. That could also be part of the response. As you said, you measured that and you couldn't find anything. You couldn't find any changes in reactive oxygen stress. But if I look at your chapter 3, I see that there's a marked upregulation of duo X2 and duo XA2, which are involved in NADPA oxidase, and NADPA oxidase causes increased production of reactive oxygen species. In addition to that, there's also a regulation of NRF2 signaling, which is also downstream of either oxidative stress or sulfur stress or nitric oxide stress. So it looks like there is oxidative stress. So I wonder, how did you measure your oxidative stress?
Yeah, so me personally, I didn't measure the oxidative stress, but I know it was measured in the media.
How did you do that?
I didn't do it. So I never worked with oxidative stress in particular. We only did gene expression. When the other opponent was asking me, we were talking about the oxygen from the culture system that can induce oxidative stress. But I think the oxidative stress that we can see in the slices is coming from the mitochondrial damage that happens during culture.
I was going to ask that, did you look at the mitochondria early after culture? Are they changed? Do they change their morphology? Is something happening there? Did you look at the genes from mitochondria? Are they changed early after culture?
Yes, our colleague Isabel Stribos looked at it, and she saw changes in kidney slices after 3 hours of incubation already. She also looked at BCL-BAC's gene expression, the markers of apoptosis and of mitochondrial degradation. She also checked the TOM20 mitochondrial receptor, and she saw changes, and she saw a time-dependent mitochondrial damage.
So the oxidative stress in the mitochondria could be a target for better preservation of your slices in the future.
Yes, and I think we should also consider the PPR alpha pathway, because we see a lot of downregulation of this pathway, and I think this can also be a target to try to compensate and have less oxidative stress and less mitochondrial damage.
Okay, thank you very much. I'm very happy with your answers, and I give the word back to the rector.
Thank you very much, Professor Van Goor, and it's then my pleasure to invite for the next opposition Dr. Nagelkerker from the Department of Pharmaceutical Analysis at this university. Dr. Nagelkerker.
Dear candidates, I would also like to congratulate you on what evidently has been a tremendous scientific journey. And just to satisfy my own curiosity, my first question, how many slices did you actually make?
Highly learned opponent. Definitely the amount of thousands, because we do have experiments that are at least 200 slices. So, for example, for chapter 4, I do remember I got to experiment 63, and I went from like at least 30 slices to 180. So
That's impressive.
yes,
That's impressive.
thousands.
So I would like to touch upon the dosing side of your studies a little bit. So if we go to page 120, that's where you use elafibranor, if I pronounce it correctly. And in your in vitro experiments, you start by adding a concentration of 1 micromolar. So I couldn't resist doing some calculations. If we assume the molecular weight of 384.489 grams per mole, and you have a tissue weight of your slice of approximately 5 milligrams, if I do the calculations, and hoping that I did that correctly, you would end up with a concentration of 100 milligrams per kilogram of tissue. Now, you also refer to in vivo experiments where the slices or where your tissue, your animal, was treated with 15 milligrams per kilogram. So that is a huge difference in dose. Also, if you consider that in the animal it gets diluted and not all the dose will go to the liver. So do you think that this issue should be addressed in your model system to improve the predictability of what you find in vitro towards in vivo?
Yes, so we did use indeed, I didn't do the calculations to see exactly the concentration, but I do believe your calculations are correct. As I mentioned before, we do have a reduction in transporters, so we do need higher concentrations of the drug. These doses were selected based on the EC50, so the effective concentration of the drug needed to activate PPR alpha. We also have a shorter time, so we want to see an effect. So we would not expose the animals to such a high concentration in vivo, where we can do the experiment for weeks. But in order to see an effect, that's why I also had like also 0.2 micromolar and 1. We usually see effects with higher concentration. So we believe that our system requires higher concentration than other models. And we also check the viability, so there is no problem of the compound damaging the cells. And for only a short time of 48 hours, I do think there's a need of higher concentration.
But then if you want to take the next step with this model, you would also still need a step intermediate before going into humans, right? Because you need to have some idea of what dose you might want to expose a patient to.
Yes, of course. So I would say the slices are one of the first preclinical tools to use to assess the efficacy of a drug, but I would not go directly into humans. I would still use in vivo models, especially for calculation of pharmacokinetics. However, I studied the effect of omeprazole in chapter 5, and this compound was also studied in human lung slices, and they did pharmacokinetic studies on lung slices, and then they extrapolated to human, and they went in phase 1 clinical trial, which was also surprising for me.
So there's definitely still potential.
So with the right calculation, with the right model of pharmacokinetics, there can be an indication, but I still think they took into consideration in vivo experiments.
And then the second question. So when you make these slices, right, you take the organ and you punch out a piece of tissue, which you then chop and put in a dish. So does that not disturb like the whole organization that the organ might have? For example, in the liver, the zonal arrangement, you lose obviously your blood vessels because they're not functional anymore. So how do you see that? Could you expand upon that a little bit? How do you see the sort of microenvironment? Is that still there, or is that completely gone?
Well, most of the microenvironment is still there, but indeed the slices are not all the same, because they are closer to bigger vessels or to smaller vessels. They are coming from different lobes that also can influence. So there are differences, and that's why we cannot assess these differences just when we do the slices, and they are randomized when we culture them. But we see differences from experiment to experiment, so we could put these differences based on the different regions where the slices were produced. However, I think it's impossible to assess a slice and try to separate them based on the area that they come. But I do believe that most of the microenvironment is there. You were talking about the blood vessels. We have a colleague that is doing studies on the contractions of the blood vessels using human slices, and indeed the contraction is still preserved, so the vessel can still react to vasoconstrictors. So although there is no blood, the blood vessels and the endothelial cells are still responsive.
Mm-hmm. And then you used RNA sequencing on the whole slice, so that's a bulk technique, if I can call it like that. So how would you then assess, if you say, well, the microenvironment is still there, but you cannot assess that with your RNA sequencing, right? So could this model system in a different way serve as, a method to assess regional differences in, for example, drug response and those kind of things?
Well, we could try to do single cell sequencing, but then this technique is quite challenging, and there would be a lot of work for it. But our studies were done to characterize all the changes that occur in our slices, in our culture condition. So this was the first step. So I think for future studies, this could be indeed investigated. But I don't know exactly how we can, well, first, yes, we have to assess exactly where the slice comes from, and then try to assess by probably single cell sequencing what changes are happening in that population of cells.
Would it also be an idea to go not into RNA sequencing, but potentially proteomics?
Well, of course. But for that, I consider that first we should be able to culture the slices for a longer period, because we noticed that the protein only, we cannot measure many protein changes in our slices in 48 hours.
Because
Ora, finita.
because it takes too long?
Because the translation takes longer, so yeah, that would be a problem. But a longer cultivation would be the answer.
I think it was said ora finita, so you can stop talking. Ladies and gentlemen, the College of Deans will now withdraw for some moments to consider its decision.